Swimming behavior is present in several microbes that live in aquatic media, from worms, sperm cells, to little photosynthetic algae. Swimming can appear as an adaptation to external stimuli, like variations in temperature or illumination. At a single cell level, motion is shown to be a good representation of behavior, which in this way can be characterized quantitatively. For instance, the beating patterns of cilia can be described as a traveling wave. When we pass to collective behavior, we observe beautiful shapes and organizations present in living systems like bird flocks or simpler systems like interacting spins. The important thing about collective behavior is that when individuals are self-propelled, like living organisms, we cannot apply the same physical laws as with non-living organisms. We can imagine, for instance, two microbes colliding, and we know that momentum is not conserved. So concepts of fluid mechanics and stochasticity must be considered to describe these motions. What we would like to do is to relate single-cell behavior to collective behavior in a quantitative way. Two mutants from the same species can show different swimming behavior. But what if now we put a lot of cells together? We should expect motion to differ between different mutants as well. But how is that motion related to the single-cell behavior? To answer this, we will use the model organism Chlamydomonas reindarti, a photosynthetic unicellular algae that swims, and we will analyze its swimming behavior. This will be done by recording their motion over time using a simple microscope that we will build, integrated with a high-quality camera. The setup will be low cost and compact, which will allow us to parallelize it to obtain a large amount of data of their positions, velocities, and density. The cells will swim inside a microfluidic device, imaged from below with an inverted microscope. Inside the main chamber, there is a trap that lifts when pressure is applied by the cell's media that is pumped in. This way, cells can get in and swim. Then, the trap goes back down and doesn't let the cells escape, allowing to record their motion. We thought about the simplest optical system that could work as a microscope with 1x magnification, since these cells are big enough to be identified. This would consist of a light source, a condenser lens to collect the rays and send them uniformly to the sample, an objective and a tube lens to create the image, and finally a camera to collect it. We have built our first prototype, which is compact, easy to mount and operate, making it practical to use in a laboratory. With this prototype, we could obtain images with good enough quality to track cells. We are now 3D printing mechanical parts for the microscope, fabricating microfluidic devices, and defining the final experimental setup so that we can parallelize it. Ultimately, we want to obtain data from different mutants of Chlamydomonas with constant environmental conditions, as well as of a certain mutant with varying environmental conditions. With these, we hope to map behavioral to environmental parameters and map single-cell behavior to collective behavior. This project is being developed at Instituto Gulbenkian de Ciência and at INESC Microsistemas e Nanotecnologias, together with my colleague Yatart Bazin and my advisors Pablo Sartori and Vânia Silvério.